It's a pleasure to have you back here for session 10, and we're here talking about mollusks in motion and different species for different reasons. You may know, and I'm sure a lot of you know, that mollusks are engineers of coastal ecosystems. They participate in the creation and modification of the ecosystem. They are also bioindicators of the status of the ecosystem, and it's quite subject to cutting-edge science that we're going to see here. So this session spans a large diversity of mollusk research, conservation as well, including the behavioral studies of octopuses in Massachusetts, revealing cognition, problem solving, and environmental interaction.
Research in Germany on color production in sea slugs, offering insights into bio-inspired design and communication in marine species, and large-scale oyster reef restoration programs in New York, combining ecosystem service delivery with urban ecological renewal.
To join us so for this panel, we are really happy to have with us Kendra Buresh, who is a researcher at the Marine Biology Laboratory in Woods Hole in Massachusetts in the USA. Also with Kendra Raphael Tremblay, a MARE student in first year, currently in immersion project at MBL. Victoria Mesich is at the Max Planck Institute in Germany, and Emma Sandrini is in New York at the Billion Oyster Project. Hello, everyone. Nice to see you from the other side of the Atlantic for some of you. And well, if you're okay, Kendra, we can start with you, addressing a little bit who you are and what's your research at MBL. The floor is yours.
Sure. Okay. Yeah. My name's Kendra Burish, and I work at the Marine Biological Laboratory in the Hanlon Lab, and we study cephalopods. And in particular, we're interested in the sensory systems of cephalopods. And we like to take a sort of a top-down approach where we start with a whole organism and from studying their behavior, both in the wild and in the lab, and then sort of go down into a finer level, looking at how they control things with their complex nervous system and their complex skin coloration system. And we are focused right now, well, what I do is mainly research in the lab, not in the field. And our lab work is really focused right now on octopuses. And we're interested in particular how the arms and suckers of octopuses work, and how the suckers are used as sensing organs. The suckers of octopuses are their main way of interacting, or one of the main ways of interacting with their environment. They have incredible vision, but they really explore their environment with their suckers. They can, using chemo and mechano-tactile sensing, they look for prey, they manipulate items in their environment to sort of create their den, to capture prey, etc. And so we're interested in how those arms work together to manipulate things in their environment, to bring the suckers to different parts of either where they're looking for prey, so getting chemical cues or tactile cues, and manipulating things with multiple arms. But we're also interested in the suckers themselves and how they use multiple suckers together to investigate things in their environment and to do all sorts of different tasks.
Thank you, Kendra. We'll also learn more with Raphael, who is working on some of these aspects right now. And before we go to you, Raphael, maybe we can have a look to your 1-minute video where we see you in the lab. So see you in 1 minute.
What if I told you octopuses could help save lives? Would you believe me? Hey, I'm Raph, an intern at the Marine Biological Laboratory in Woods Hole, Cape Cod. And I study octopuses, more specifically their fascinating suckers, to help inspire soft robotics. My days are spent in the wet lab caring for animals and filming how they use their suckers. I study the sequence in which suckers attach, looking for patterns in different arm regions, distal, medial, and proximal. By studying how octopuses use their suckers, we're helping engineers design soft robots. This is where the life-saving part comes in. These robots could assist in delicate surgeries, explore dangerous underwater environments, or deliver medicine in disaster zones. So now you know. Beyond being incredibly smart, octopuses can also inspire life-changing technologies.
So, Raphael, can you tell us a little bit more about what you're doing in your experience?
So as Kendra mentioned, they're looking at the octopus arms and suckers. So I'm more specifically looking at their sucker recruitment. So it's when one sucker attaches, in which sequence do the following suckers attach. And more specifically, my experiment is looking at the different parts of the arm. So as I mentioned in the video, we kind of have the arm in 3 different parts. And I'm looking to compare the recruitment patterns in those 3 different arm sections. As like the suckers in the proximal part of the arm are much bigger and are recruited in different ways already based on observations. Are recruited differently than the tip of the arm is, where the suckers are much smaller. And also, as I mentioned in the video, this is used for soft robotics, which is different than hard robotics, which you think of like a robot being made out of hard material. Soft robotics puts an emphasis on flexible robots using soft materials. So what better than the octopus as an inspiration for this? I mean, they're invertebrates. They're incredibly flexible. Their arms can do really fascinating things, twist in all these directions. So that's kind of the goal of the research that I'm doing.
Thank you, Raphael. Can you just precise a little bit what you mean by recruitment for the audience?
Yeah, so recruitment is, well, I don't know if you can see it well in the videos, but we're looking at Agro's disc. We use Agro's disc, and the octopus will come up and attach one sucker, and then we'll kind of wrap their arm around it, so they'll recruit basically the following suckers. So it's a recruitment of the sucker, so which one attaches and in what sequence do the following suckers attach.
Okay. So thank you, Kendra. Thank you, Raphael. We are going to come back to you for more exchange and questions. We may go to you, Victoria. Victoria, you're in Germany. Actually, you were working on octopuses last year in Italy, so maybe we can jump into the discussion about octopuses after. But now you're working on sea slugs, other fantastic creatures, and the ways they're making colors. So you also have a short video, but we will use this video as you're explaining what you're doing. So, Victoria, can you explain to us what you're doing in Germany?
Yeah, sure. So I am Victoria. I'm a second-year master student at MarWorld, and so in Germany I'm doing my master thesis, and we are studying a species of sea slugs called the Lysia crispata, and you will see in the video how it looks. And so we are studying the production of the colors of the sea slugs, and we want to understand how they produce the colors and to understand also the biological role of these colors. Thanks to this study, we can create some bio-inspired materials that can maybe explain after the video, because it will be more clear to understand.
So if you want to click on play and continue to explore your research at the same time?
Of course. Do you have the sound?
No. So no, there is no sound, I think.
Okay, so I will put this on. Okay, should.
I think, Victoria, the sound is not connected to the stream from your ends. Can you just explain the video?
Is it working now?
No, just can you continue and explain the video?
Sure. Right. Okay, so I will start again. Okay, so I want to explain how to be a marine biologist, but without being at sea, but be in the lab. So to find the supermarine organism is the little sea slugs, the Lysia crispata, and it looks like a coral, but it's not. And so it belongs to the class Gastropoda and the order Sacoglossa. And so these slugs, they have a super interesting ability, is to suck out the chloroplast directly on the algae they feed on, and so thanks to that, they are able to do photosynthesis because they have the chloroplast in their body, as we can see with the green spot in their body. And so we found a characteristic that hasn't been studied yet it's the color of the sea slugs. So they look like salad, but they have really various color, but mostly blue, as we can see. And so those colors are produced not by pigments, but by structural coloration. And so the structural coloration is the refraction of the light depending on structures in the tissue, and so it's different from the pigments. And so we try to understand how is it produced, this structural coloration in the slugs. And so we could see a bit in the video, but it includes lab works and also a lot of computer works.
Okay. Thank you, Victoria. We're coming back to that in a few minutes. And so we'll jump now to New York. I don't know if it's New York City or New York State for now. Emma, you're working in a restoration project. I don't think we can imagine more urban than your restoration project. You're going to explain that very soon. We are changing also the species. We'll learn first more about directly a video, and we're coming back to you.
Emma. I've been working at the Billion Oyster Project as a field restoration technician for almost a year now. The Billion Oyster Project is an environmental NGO dedicated to restoring oyster reefs in New York Harbor by involving local communities and students. In my role, I'm involved in a wide range of activities, from fieldwork and monitoring to data analysis and developing protocols. In this video, I'm taking you along to Brooklyn Bridge Park, home to one of our largest reef installations. We placed 75 reef balls there in August 2024, and for the past 8 months, oysters have been growing and developing on them. This week, we are monitoring selected and random reef balls. We start by lifting each reef ball out of the water. Once on board, we photograph randomly selected quadrats on the reef ball surface to document the epibentic community. Next, we carefully examine each quadrat, counting and measuring the height of both live and dead oysters. Most of the oysters we are seeing are still juvenile, under 20 millimeters in height, since they settled less than a year ago. We are also documenting broader biodiversity around the reef balls, measuring the lengths of each fish, counting invertebrates and other critters that show up on the deck before returning them to the water. All the data we collect will be entered, analyzed, and reported to help us better understand how oysters are growing at this site, and hopefully they'll continue to thrive and reproduce for many years to come.
Thank you. Thank you, Emma. Do you want to follow up on this video and explain a little bit more what you're doing and why you're now, how you've been able to jump into New York from Paris?
Yeah, so hi everyone, I'm Emma. I'm a Morris alumni. I graduated last year, and I'm very lucky that I found a great job at the Billion Oyster Project, which is an environmental NGO. And as I explained in my video, I'm a restoration field technician, so I work a lot on the field, working with data afterward. And yeah, Billion Oyster Project is one of the few initiatives to protect the marine environment in New York City. So we work directly in New York City, as you've seen with the video, all across the 5 boroughs since 2014. We have now 18 restoration sites, and the big goal is to install 1 billion oysters by 2035 to have a sustainable reproduction of oysters. A little fun fact is that in 1600, so a long time ago, there was more than 220,000 acres of oysters, and New York wasn't called the Big Apple, it was called the Big Oysters, because oysters were the main source of protein for indigenous communities. And with industrialization and overconsumption, they all disappeared, and the ecosystem that goes with it. So our goal in the Billion Oyster Project is to really bring back the substrate for the oyster to grow on and the population, to recreate the sustainable oyster reef, sustainable reproduction of the reef, to have a better water quality, because the oyster is a filter feeder, so it can filter up to 190 liters of water per day. So it's a big amount, and like capture all the pollutants, and it can allow to like create a complex ecosystem to provide food and habitat for a lot of different species, marine species, and also it can protect the coasts from erosion as well. So yeah,
a lot of ecosystem services.
Yeah.
Back to that. So can you tell us a bit about the funding for example? It's a big project. So who is funding that? Is it like the city or Messen?
So our main source of funding, the funding comes a lot from private sources, but we also receive funding from the city and state of New York, and as well from like the federal government, which. Yeah.
And so just before we're pouring maybe the questions for everyone, you're in a car because you're actually on the field.
Yes, I wanted to mention I'm actually on the field, like right where we saw all those videos. We also have a smaller nursery, and today we're working with a local community to monitor one of our reefs there. So I'm on the car for the call.
Great.
Yeah, so I might just open up the questions maybe to the working on octopuses and sea slugs. And I was wondering, what are some of the challenges with working with these animals in the lab? And what are some of the more surprising experiences you've had during your kind of like your research and kind of like experiments? I don't know, maybe if Raphael and Kendra want to take it first.
Yeah. Yeah, well, I mean, doing behavioral research, you're working with live animals that don't necessarily want to do what you want them to do, or will do something you never imagined them to do. So I think a lot of it, I mean, since the start of my internship, has been kind of like adapting the experimental design and adapting the experiment as it goes also. To adapt to how the animal is reacting. And then again, maybe one animal individual reacts well to the experiment, and then another one just doesn't want to do it at all. So I think that's kind of a challenge. I don't know if you have something to add.
Yeah, we were just having a discussion about this the other day with some other students who are in the lab about how when you're designing these sorts of experiments, you have to try to think a different organism so that you can design an experiment that's going to answer a question that you're interested in answering. And you can't necessarily approach, as you know now, but like you can't approach the question from the point of view of a human all the time. You have to try to imagine what it would be like to be this other organism in some sense, to be able to answer or ask the right questions and set up your experimental protocol so that it'll work. And even still, well, that's really impossible. But you know, we try our best, and then with we just have to always modify experimental protocols as we go on until you actually settle on something that's going to work. And we work with squid, cuttlefish, and octopus here in the lab. Raph is working with octopus. They're like the most difficult of those three groups to work with because they just are very curious, and that sort of gets in the way a lot of the time.
In the way. Okay. Coming back to you, Raphael, what's a normal day of an intern? Like, I guess there is a lot of work to do also to maintain the organisms.
Yeah, so a big part of my my job here is taking care of the organisms that we have. As Kendra said, we don't just have octopuses; we also have cuttlefish. So a lot of, I'd say half my time is spent cleaning and feeding the organisms that we have, and then the second half is running my experiments and doing a lot of video analysis, which is less fun. But very crucial and very important to the research. So I'd say it's interesting also to interact with the animals in a cleaning perspective, because you kind of get to see the animal act in the way that it would act differently than in the experimental context. And I feel like getting the experience to take care of the animals kind of like shaped how I set up the experiment and shaped how I'm interpreting the results as well. Yeah.
Yeah, you learn a lot about animals, taking care of them. Yeah.
Mm-hmm.
Thank you. Another question for you, it's about the sample size of your research. I guess the sample size of research working on octopuses is not the same than when you were working on sardines in the water. So how do you deal with that? How many octopuses are you comparing for following?
Yeah. Do you want to start answering, or I can?
No, go ahead.
Yeah, so right now for my experiments, we have a sample size of 5 octopuses, just because of like what we decided to look at and the time constraints and all that. Yeah, for sure, the sample size is smaller. And like I said, not all of the octopuses want to partake in the experiment either. And so that's kind of part of like adapting it, is figuring out, setting maybe a lower sample size, and then getting to try out different individuals and see which one responds well to the experiment.
Yeah, so I guess I would just add that in animal behavior in general, that it's often you see small sample sizes because it's difficult to run live animal behavioral experiments, as Raf said. So we'll like a good sample size for us for most experiments is somewhere between 5 and 10 animals, which I know is a really low sample size for a lot of science, but that's pretty well accepted in the field that we work in.
Of course, and there's also the ethical point of view on that, so that we are not going to follow tens and tens of organisms at the same time. Thank you. Thomas, do you want to continue?
Yeah, I have a question for Victoria. As you know, you've worked with, last year you were working with octopus, and this year you're working with sea slugs. Like, what are the differences you've encountered between these different projects?
Well, the organisms are really different, and as Raphael and Kendra said, the octopus are super curious, and also it was behavioral analysis too, so it requires a lot of patience and adaptation, like they say too. The octopus doesn't want to cooperate always, but for the sea slugs, I'm doing mostly optical and microscope images, so they don't really have a choice to cooperate, let's say. And, yeah, they are not as I don't know how to say. Well, they are more calm than the octopus, so that would say the biggest difference. Is easier to work with the slugs than the octopus for sure. But also one of the difficulty is we have to adapt to the health state of the animals, and this is a big challenge. That if, for example, if the animals died, then we have to get more samples, but it's difficult to get, and we have to adapt to all that.
Of course, I'm sure you make the best of your efforts for them not to die, and
yeah, no, of course.
always unfortunate. Can you come back also, Victoria, on the bio-inspiration aspect of your work and what could be expected from your research?
Yeah, sure. So the structural coloration is studied a lot in different organisms, but mostly terrestrial, less in marine organisms. But also in the lab, they are also studied structural color on algae and in bacteria for living organisms. And the goal is to study how they produce those colors in those organisms to get inspired. Like the further goal is to create some dye and paints that are more sustainable materials than the actual ones, because the actual ones that have very toxic compounds inside, so they want to have natural color. Also, thanks to structural color, it will be more sustainable in time, and the color will be very vivid and better. It will not degrade with the sun.
Lasting longer, okay.
Yeah, it will stay longer than pigments that are used in the dye for now. And another aspect is that it could be interesting to understand, this is not about the area intervention, but most biological aspects. It could be cool in further study to understand the link between the fact that they could integrate chloroplast and the production of colors, for example, if there is a link between these processes or not.
Thank you, Victoria.
Yeah, I have a question for Emma. I'm really curious to know about some of the challenges of working on an ecological restoration project in like an environment like Manhattan. Just like kind of like, how do you negotiate that? What kind of like hurdles do you encounter, and how do you try to resolve and navigate them?
Yes, so that's a great question. So working on like ecological restoration in urban environment, I think it like you face a lot of challenges because I don't know if many of you have been to New York, but it's not the cleanest city on earth. Like it's very polluted. The waters are very polluted. So and as so yeah, this is like a first challenge for the oyster to grow because its lifespan will be way more shorter than when it goes in like the pristine water, like I don't know, in like the Bassin d'Arcachon in France, for example. I hope you can hear me well and that I'm not cutting off.
Perfect.
Okay, cool. And also pyramid-wise, so since we're like working on a large-scale restoration project, it's we're like accelerating every year the amount of oyster we're installing in the water, and the pyramid
getting a little bit more difficult. We'll see if Emma is coming back. Maybe we can jump back to Emma in a second. Anyway, we are going closer to the end. So what I would like to propose you is maybe taking the time in a minute. Sorry, Emma, you were cutting.
I'm really sorry.
No. If you want, you can finish your sentences.
Yeah, just so like we're like the first to do like the big scale restoration in the New York City, and yeah, it's challenging, and we're learning each step of the way to like to keep going and learning a lot.
And so, have you tried putting your head in the water in New York City?
I'm sorry?
If you try putting your head underwater in New York City.
So we have a team of diver, actually. Like we have very strict rules and health protocol for them to dive because after like big rainfall, the like overflow of the sewage, not the best to dive. But yeah, we have strict protocol, but the divers help us a lot to access our reef that are like a bit more deeper in the water. But we have a lot of reef that can be accessible from shores with a good pair of waders, and so we're often in the water, but
I saw that in your video. Amazing. I can't imagine, once again, like you're living in New York City, but you're by the sea. We were just talking about that before the live. So it's really a special feeling of living in New York City, doing that. I'm not sure there is so many New Yorkers being so connected to the ocean. That's what you're doing.
That's something we also want to like really have with the Billion Oyster Project, is this connection, like include the community and the students, like education initiative and community science to really promote this connection to the waterways, because New York is like a coastal city and a lot of New Yorkers doesn't see it that way, even like tourists. And from the way I've seen it, being on boats, being always on the shore, it's where New York is the most appreciable, I would say. So yeah, it's really great to work with a lot of different people with a lot of different background. And
thank you. I'm happy you've been able to stay until that to say that. Raphael, Kendra, Victoria, Emma, we're going to the end. Let's take the time to give a kind of take-home message to our audience, something like in 30 seconds. What would you like to tell our audience? We can try with you, Victorias and Emma.
Yeah, sure. I would say that it's nice to be able to work with marine organisms, even if it's in the lab, and that even if it's sometimes sad to work and to maybe hurt the animals. It's still interesting to protect them and to learn about them and to be able to protect the population as
a better understanding of the population. Emma, and then we're going to Raphael and Kendra.
Yeah, I would say I would encourage a lot of like people to like be in connection with the waterways in their neighborhood, because I think all over the world there are a lot of initiatives to promote this connection with the people to their waterways, and you can be engaged in different activities, learn a lot from like scientific aspect of different like restoration method happening. So yeah, I would encourage everyone to look for this organization or infrastructure that organize those things in your neighborhood and participate in it, because you'll be surprised to meet a lot of amazing people and learn a lot about the ocean.
Yeah, it's true. We're talking about that just last session. It was about education and outreach. It was a lot also in Central Caribbean, but we can do that everywhere. Thank you for sharing, Raphael.
Yeah, I would say staying curious. There's a lot of, I feel like most ideas and decisions come from curiosity, and there's a lot to be inspired from in the ocean, a lot to learn from marine organisms. So to stay open and curious.
Thank you, Raphael. And Kendra?
I just want to say that how much we've enjoyed having Raph here in the lab and how amazing it's been to have her here with us. And she's done such a great job. And also staying curious is, I'm just going to sort of riff off of that, which is that having students come here in the lab, especially when they're like Raph, who is just so into the research and is so thinking things through so much, it really helps us to stay curious, because we have students coming through who are so curious, and it gives like a nice fresh perspective. Wow. So I guess my point is it's good to get lots of different perspectives from people of different groups and backgrounds.
Well, no pressure, Raphael. Thank you a lot. And so, Raphael, so thanks a lot for all of you. It has been a pleasure having this session with you. I hope you will continue to watch the coming sessions, because we are going to continue. The next session, so in 30 minutes, session 11, will be on plastic, with some aspects of course, the issues, but also alternatives developed in our labs, and ideas, of course, to collect and give a second life to plastic. Thank you a lot to all of you, and yeah, see you soon. Bye.